I was in the wrong room, you guys. My bad. Hello. Hi, Dr. Vidal. How are you?
How do you go into the wrong room for your own Zoom talk?
No, folks were using the same link from this morning or something, and then I thought it was the reminder that I have to log in and I just hit start. I'm
Ah,
not that, well, I mean, I've been having issues with all everywhere. So, but that just means my classes have been very interactive, so that's a good thing.
yeah.
Okay, I may or may not just because typically Dr. Brown and Dr. Bannon do these reviews, and they pretty much just do sort of like from beginning to end as far as going over all the content again. And those tend to sort of run the length of full 2 days worth of exam of classes. So sometimes they last like 4 hours or more. But I encourage you to actually, if you have specific questions, um, I'm gonna start just going through the topics that the learning objectives in our syllabi would address. And so as I'm going through that, if you guys have any questions, feel free to just ask. If not, I'm gonna bounce back and forth depending on my what do you call it, stream of consciousness, where it takes me. All right.
And if you'd like, if you'd also like, if you want to put it in the chat, I can help out so that way you can keep talking.
Yes, and I appreciate Dr. Vidal's going to be here to not freezing for me. I'm sorry, Miriam,
Try logging out and
but
go back in.
but maybe I'm frozen for you. But I haven't brought up any of the slides yet. So how do you usually do these, or just you just do the Kahoots? We played the Kahoots with the first years yesterday, and that was fun.
Okay, good.
Yes, I think it was a confidence booster for the mo for most people. I can't speak for everybody, but did the students? Oh, am I not projecting? Yeah.
Yeah.
Oh, that's weird that it's not. Oh, I'm here. I don't know why. You guys can see my face, no?
I can't, but apparently some people.
Oh, maybe let me just log in and off again because I think it
Maybe your video
probably
is in
thinks
the
I'm
other
still
room.
on the other side. All right, BRB.
All right. Yeah, so if you have a pressing question and you'd like to ask Dr. Malver, that's fine. If not, please put it in the chat and I will try and keep up with questions in chat as they come.
Hi, Dr. Vidal. It seems that the Zoom, I think, is at max capacity because some people are having trouble getting in and getting a message saying that the max capacity has been reached.
Oh no. Okay. Was
Uh,
there a way to deal with this, Luis?
uh, no, not really, because we got capacity downgraded after COVID. The only thing I could tell you is if you could, if the only thing I can say is if you are
This
all in
is
the same
recorded.
spot, if you are all in the same spot, you can try and just group up, right? So if there's a bunch of you, I don't know, in Harlem, if there's a bunch of you that are in LH1 or something, if you wouldn't mind designate somebody into one laptop, gather around, and that way we might free up some login spots. Thank you, Lauren. Appreciate it.
Nice. Thank you so much.
Yeah, so to the extent possible, and for Middletown as well, because I know if you guys, I know you guys, those of you that are in the dorms, if you can, if you don't mind. Hey, listen, it's a little party. Group up together and get cozy around one laptop. It's a good way to make friends.
Yeah, and Harley's like, do you want to run the cahoots? Because that might be a nice icebreaker. Um, but I mean, we could just start and talk and just hang out, chill. Okay? All right. Let me just put you guys in the smaller window where I'm actually facing y'all. And then we're screen two. Okay. So let's see if I can get my chat box somewhere else. Nice. Okay. So the first week's topics included the cell, pretty much, cytoplasm, nucleus, and important to know the histologic techniques that we use to be able to highlight different features of the cell. If you're interested in certain proteins, what techniques might we use to bring out those particular proteins of interest? Yes, Melanie, we are doing topics 1 and 2, pretty much the two classes that you've had in the last 2 weeks. Impossible to test you guys on week 3 because that class is going to happen after Monday, at least for us. When do you guys do week 3?
It's week 3 material, yeah.
Okay. So the plasma membrane, as you know, is a bilayer, lipid bilayer, where there are hydrophilic surface as it faces outside, but also the layer that's also hydrophilic, so it likes aqueous or polar surfaces, would face the inside as well. Then there are hydrophobic center where the hydrophobic tails of each of the layer face each other. So pretty much oil mixes with oil, water repels oil, and so those two sides are on opposite sides, and therefore that's how the plasma membrane maintains its bilayer configuration. Inserted between these molecules, these phospholipids are different proteins. And as you know, cells have to be very flexible. And the components of the plasma membrane vary. Sometimes it varies in composition of how much cholesterol is inserted in there, depending on the temperature. So, and the configuration of the plasma membrane, some folks have, we can highlight. Yes, we are going to talk about special stains as well. I mean that, we can jump into that. That's cool. Um, I guess I could just finish my point. So plasma membrane, different proteins, some lipids, cholesterol are also inserted in there, and it can vary composition depending on temperature, whatnot. So, and cholesterols, think about them as sort of hinges that allow for the fluidity of the plasma membrane to vary. This is just a depiction, an initial image, the first image that we're looking at right now. Is this a transmission or a scanning electron microscopy image? Yes, it's transmission. Really, does the electron pass through the different parts of the cell? And if something looks dark, that means it's really hard for that electron to go through. So we would call those electron dense. And if it's a light area, that means the electron is able to go through. That's electrolucent. Okay. Here's sort of the fluid mosaic model. These little proteins are sort of islands that are just floating in a sea of phospholipids. Is this a scanning electron microscopy image or TEM? Yes, surface scanning. Yes, it's more of a 3D. We're not looking through it, but rather the surface texture of the plasma membrane. And what happens is, as you know, the plasma membrane has 2 layers, and they use sort of like this wedge to separate the 2 layers apart. And what you're seeing are going to be the P phase, which You need to see how it's much, much more bumpy to see more of the proteins. And then E phase, think it's extracellular or external. Think about it in that sense. So the last two images is really to drive in the point that transmission electron microscopy is 2D, two-dimensional, whereas surface is more, three-dimensional. What is this? I'm gonna mix up stuff. So we learned this today. How do you know it's a microvillus or a picture of microvilli? How do you know that? Based on the text. Good. So microvilli are covered with these carbs called glycocalyx. And what might be a special stain that we would employ to highlight or pick up structures in the cell that have carbohydrates? Excellent. A lot of you are saying the periodic acid shift stain or PAS. And one of my favorite images are of, well, I can show that to you guys later. Diabetic nephrosclerosis. So the changes that glomeruli go through as they scar over time. And scar are full of collagen, okay? And collagen are also part of that scar are some carbs in a sense mixed up. Basement membrane is also laminins. Laminins have carbs. So in a PAS stain, it's a very good stain to highlight basement membranes, to highlight brush borders that are really carbohydrates, the glycocalyx covering the brush borders or the microvilli. What else might PAS highlight in, say, respiratory epithelium? Oh, these are just lecture slides. It's been posted, Michelle. Yes, goblet cells. Goblet cells make what? Mucins. Good. And mucins are made of what? My shortcut is carbs, but there's going to be saccharides, polysaccharides, and there's definitely beyond the scope of this class, different types of mucins. And so PAS is one of the stains that we use, special stains to highlight goblet cell mucins. There are some goblet cells that may not get picked up by a PAS stain, so we use different types of other mucin stains like the Alcyon blue stains. Those would look blue, but what is the color of, carbohydrate filled organs? I mean, sorry, not organs, but like structures or the glycocalyx. What would that look like on a PAS stain? Good. Dark magenta purplish, or depending on the contrast of whoever's taking the photo. Um, Sometimes they would manipulate it so it looks extra bright pink, but yeah, it's much, much pinker than a regular eosin stain on an H&E. Okay? All right. So on an H&E stain, how might these goblet cells look like? Yes, they look pale. And really, depending on which lab is performing the staining, most of the time, because they want to cut back on the usage of some stains, uh, you know, not as much hematoxylin, which is our typical purple dye. These goblet cells would look, for the most part, pale white. Okay, but if there's a little bit, they can afford a little bit of the H. Sometimes they do look pale blue, very, very light blue. But for the most part, they would look pale or white. And they are called goblet cells. Why? Yeah, have you guys seen a goblet? I don't have a goblet here, but a goblet that we use to drink Pinot, right?
Ye
Like
olde.
pinocytosis, the take up of fluid from the outside. Sorry, that's how I'm mixing up. Extracellular intake of fluid is pinocytosis with a goblet. All right, good. So if you don't know what a goblet looks like, go ahead and do a quick Google search on that. Okay, So let's stay on this image. So microvilli, which type of epithelium do you see them on? So intestinal epithelium, okay, where else? In kidneys, where in the kidneys? So first off, what is the purpose or what is the purpose of the microvilli, the special apical specialization of these particular cells? Good for absorption, reabsorption. So really these are extra outpouching, but finger-like outpouching of the plasma membrane to increase the surface area where transport proteins actually, or even channels can stick on so that there is absorption, extra absorption. We can increase the surface area of absorption that happens in parts of the body that have these types of epithelium. So small intestine is very, very famous for absorption of nutrients. Um, some folks said the kidneys. Where in the kidneys might we do some absorption or reabsorption? Good, the proximal convoluted tubule. And guess what? When you slap on a PAS stain on a small intestinal epithelium and proximal convoluted tubule tissue, what might the brush border or the microvilli, how would they look like on a slide? PAS stain, brush border. Yeah, it would look like you took a magenta pen and sort of colored up the tops of these epithelium because um, those would be the microvilli that have the covering of the glycocalyx on top of them. So both in the small intestine, but also in the proximal convoluted tubule, as well as the distal convoluted tubule, but not as present they're not as dense in number. So it's not as prominent if you try to do a PAS stain. Microvilli are still there, but not as voluminous as they are in the proximal convoluted tubule. Okay. So we are looking at microvilli. What might be an exam question that we could ask you? What else have we discussed in terms of microvilli, at least this week? Why do you think they can stand up like that? Yeah, what might be, uh, what is making up its internal or its core structure, and how are they staying anchored in the apical surface of the cell? Yeah, so the core structure of microvilli are made of what? Good. I see people saying actin. And how are they anchored to the epithelial cell or the tops or the apical portion of, say, the small intestine, the enterocyte? Through the terminal web. Okay. And what are the terminal webs made of? Okay, I see people saying actin. Do everyone else agree? Do you guys agree? All right. For the purpose of this class, we are differentiating or we are distinguishing microvilli and the other types of apical specialization from each other by virtue of how they look like, where they are located, what they are made of, okay, and their function. So you guys mentioned microvilli are for absorption. Their cores are made of actin filaments. They are anchored down to the cell, to the apical portion of the cell, through an actin structure called the terminal web. Okay. What are other special or apical specializations have you guys learned of this week or read about? Cilia. What are cilia, or specifically motile cilia? What are they made of? Or at least what are the cores of cilia made of? Good, microtubules. How are they anchored to the cell? They are anchored via little microtubules also. They're called basal bodies. Okay. Basal bodies would be the anchoring structure of cilia, of motile cilia. And when we say 9 plus 2, we are talking about the 9 pairs, the 9 doublets in the periphery of an axoneme. Those are 9 pairs of microtubules and a central pair. And that is the configuration of a motile cilia. Okay. And they also have these dynein arms. And I've demonstrated this because I've never forgotten this. I took a cell biology class back in 2005, so 20 years ago. And I had the cell bio teacher just told us, look at me, these are my dynein arms, and this is how they move, against each other. And that's how they produce from very far away a unified movement. The cilia should always beat in the same direction. Okay. All right, microtubules. So just to emphasize the 9 plus 2 configuration of microtubules within the axonemes of cilia, that is specific to microtubules. Okay, where else do you see microtubules? So Nathan, microvilli do not have basal bodies. Microvilli are anchored via terminal web, which are made of actin as well. The cores of microvilli are made with actin, as well as its anchoring structure, which are the terminal web. Okay, the question was, where
Thank you.
else do you see microtubules? Yes, the spindle apparatus during mitosis. Um, every time you see it looks like, you guys know what a spindle is? Like anybody sew or do knitting? Anyways, so they just look like a spool or a spindle. And when they are pulling apart the sister chromatids, what's happening is actually these microtubules are depolymerizing. They are becoming shorter by taking off little pieces of tubulin out of, their structure, so they get shorter. What anchors the spindle apparatus on each end of what will end up being its own respective cell? The centrioles. What are centrioles made of? Awesome, microtubules. And guess what is the configuration of centrioles? They are not 9 plus 2 because the 9 plus 2 are reserved for motile cilia. Yes, there are 9 triplets in the circle. 9 triplets. Okay. So, all right. We talked about microvilli. Purpose is absorption or reabsorption. Cores are made of actin, anchored down onto the apical portion of the cell through an actin structure called the terminal web. Versus versus cilia, motile cilia will have a 9 plus 2 configuration of microtubules in its axoneme anchored onto the cell via basal body. Okay. And the movement means the purpose of a motile cilia is what? Because it's motile. So it moves, it propels stuff. Where might you find motile cilia? So the conducting portion of the respiratory tract will have motile cilia. It would not be the gas exchange component in the very periphery of the lungs. That's not where motile cilia would be. It would be up on the bronchioles, in the bronchi, the trachea. Okay. That's also where do you see these cells that have motile cilia? They're also, some of their neighbors are what? Cells that produce mucus, right? So not only are they next to goblet cells that are producing mucus, what's the purpose of mucus in the respiratory tract? And you could correct us, Dr. Vidal, or me too, if you want. I will not necessarily see a private message if I'm misspeaking, but
Which
I will correct myself.
oh, for what? For the purpose of mucus in the respiratory system?
No, for anything.
Oh, no, no.
Yeah. Okay. Sure. Lubrication or even humidifying the airway, right? You guys ever had dry air and how uncomfortable it is to breathe with really dry air outside? Sort of, we want to humidify the respiratory tract so it's, more pleasant to breathe. It also helps, well, I guess because there are cells there, it's gonna humidify the respiratory tract. And yes, to trap the debris. And you don't want that trapped debris with the mucus stuck there. So the cilia are there to propel it out. You can hawk it up in the form of loogie or snot, whatnot. If there's a lot of it.
That's a medical term, isn't it?
What?
That's
Hawk?
a proper medical term, a loogie.
Sure.
Okay, just
Sputum, you can do hawk up a sputum if it's really deep down there, your mucus secretions in your nose. So, I mean, I know there are some products out there that they use the word mucinex. It's supposed to break up the mucin so it's easier to expel or expectorate. All right? Which is the medical term or the scientific term for hawking. Expectoration. And that was part of the lyrics in one of the songs in Beauty and the Beast. Okay. All right, moving on. I'm gonna be stuck on this because this is, I can do this review without slides. But so we talked about microtubules. Motile are part of motile cilia, but also microtubules as part of the spindle apparatus and the centrioles, the anchoring centrioles of the spindle apparatus during mitosis. Where else do you find them in the cell? I know we're still on the glycocalyx slide. I want you guys to not even, I'm going to turn off the sharing. I'm just going to talk to you guys. So, where else do we find microtubules? Do you guys remember that picture? Let me find that picture. It was a fluorescent image, and there was like a blue oval in the middle, and there were these green things emanating of the out of the oval, that blue oval, and then there's this like red orange stuff that was complete, sort of is making an arc or sort of following the shape of the cytoplasm. Do you guys remember that? What was the, I'm looking at it right now. I'm looking for the slide right now.
that's in the first lab PowerPoint.
First lab PowerPoint. Okay, thank you. All right.
They've got it. They got the right answers in the chat there.
You guys remember those are why does it look like that? Why do they look like they're radiating out from, near the nucleus and out seemingly like towards the periphery of the cell? Because and you say those are microtubules. Why do they look like that? They're not undergoing mitosis for sure. There we go. I just have to remember to share. Remember this image? And you guys said the answer is what? Is E in this possibly? Yes? Sorry. Stop looking at the chat.
They got it. They're mentioning it's for cellular transport.
Good. Yeah, because microtubules serve as the roads or the railroad tracks. That's how the cell knows, like, where am I going to put this protein for secretion? Oh, I got to follow these railroad tracks because that's the direction I have to go is out of the cell. Okay. In certain disease states like reactivation of varicella zoster, people getting shingles, which means they initially were infected with chickenpox back in their youth. And when they're older, perhaps their immune system was down, the virus got, reactivated. So that virus would chill in the dorsal root ganglion. And then when it gets reactivated, it's going to move out from the nerve and out to the skin again.
Dr.
And they know how to move that way because of they're following the tracks of microtubules.
Could you let me share really quick something?
Yes.
I just want to show them because I mentioned this.
Yes, Tyler, red is actin.
I mentioned this in my class. This was a gift that I was talking to people about, but this is pretty much, uh, you need to, okay, there you go. So this is the gift that I was talking about in my class. So this is dynein
Oh,
moving,
yeah.
uh, a vesicle in the nervous system. And so it's walking where it's walking, those are the microtubules. And so you can see the protein literally walks on that rail of microtubules to facilitate the movement. In this case, it's a synaptic vesicle. So that's
Mm.
dopamine. That's supposed to be dopamine in the brain. Well, strutting, walking,
Sure.
moving.
Trucking, whatever you want to use. Sauntering.
Yeah.
Yes. Yeah, so all right. And in this image, the cytoskeletal component that's actually red is what? Someone said it. Gallivanting is good. Yes, actin. What's another name for actin?
There it is.
Microfilaments. Good. Okay. Let me see. Where am I? Important slide. Okay. We'll talk about some intermediate filaments, at least briefly, some of them with regard to the junctional complexes. This again is just a cartoon to describe the distribution. This is a two-dimensional cartoon, okay? Um, and there are many different types of intermediate filaments, and also depends where you are in the body or what type of cell you are. And this is just a nice list of, these types of intermediate filaments. Lamin
Dr.
is what,
Alvar, we don't see anything. I don't think you're sharing your screen.
okay, my bad, my bad. So this nice list here, mark it. Very nice slide, a nice table on the different types of intermediate filaments and their general locations or which cells they are in. So lamins are the intermediate filaments that pretty much holds up the structure of the nucleus. Remember, lamin is different from laminin. Where have you heard laminin? Where did you hear that's located in? Where's the location of laminin? But I just want to make sure you guys know that lamin is different from laminin, okay? Lamin is part of or bakes up the structure, provides structure for the nucleus. Okay. And excellent, Kriskaya. Laminin is part of the base membrane, and that's actually one of the components that have carbohydrates that PAS stain would actually latch onto. Okay. Nice 9+2 configuration of a motile cilia. And you can see this, the basal body here is gonna be made of 9 triplets. Centrioles, as well as centrosomes, are also composed of the 9 triplets. These guys are just anchoring, just as basal bodies are anchoring microtubular structures. That's why they have the 9 triplets configuration. But motile cilia will have 9+2. Where else do you see this 9+2 configuration? Apart from cilia. What else move? Yes, flagella. Where do you find flagella? All right. Spermatozoa have flagella. Good. So we've talked about two of the three apical specializations so far that so microvilli and cilia. The other one is stereocilia. Now, is stereocilia motile? Right. So they are not motile. And I saw earlier, I'm not ignoring you. Someone mentioned, oh, well, stereocilia is actually, we also see them in the ear. So first off, I know it has a similar name, but stereocilia are not motile. Why is that? What are the cores of of stereocilia? What are they made of?
There you go. They got it.
All right. Okay. This part not gonna be on the test. So that's a freebie. Don't study it. What anchors stereocilia to the cell? What do you call that? You may not have it, but it's called the cuticular plate. Who is Erzin? I don't know what that is. Yeah, don't worry about that. Um, but essentially, well, what is the purpose of stereocilia? At least one of the purposes of stereocilia. And you will get a little bit more of this when we talk about the male reproductive system in the second semester. Yes, it's for absorption. Pretty much there are these cells in the seminiferous tubules that are the support cells for the development of spermatozoa, and they produce this fluid, all right? And if you see them looking like as if they're moving, they're not moving, they're just going with the flow of the fluid, all right? And they reabsorb excess fluid. So stereocilia, just like microvilli, one of their purposes is to absorb. And just like microvilli, the cores are made with actin. Okay. And in general, stereocilia are much, much longer than cilia. So think about stereocilia as having shoulder length hair. Microvilli will be buzz cut, but very, very dense, tight, short buzz cut. And then cilia is maybe, has some bangs, okay? Where you can actually see a little bit of spacing in between the cilia. All right. What else is important about that? Yes, some folks had mentioned, well, why stereocilia in the ear? Well, stereocilia in the ear, they are they're apical specializations of hair cells, which are they pretty much respond to the changes and the vibration of the fluid in the inner ear. And those changes are actually impulses that are taken up by the hair cells. It's called hair cells just because they have the sericilia and transmitted through these nerves that communicate to cranial nerve 8 that is crucial in hearing, but also in your sense of balance. So they're also receptors or sensory receptors in a sense. Yes. So one of the things that I recommended, or at least this was a question that I had that was asked by one of my students, one of the master students last year, who succeeded and was able to, be one of our DOs this year. Well, how can I tell them apart? Because, it's a little confusing, right? So one of the things that I would, can you see this? No, I'm not sharing. Um, one of the things that I told him was, well, if you're confused, why don't you do a side by side? Get images of these types of epithelial examples and stick them out side by side just to visualize like how are they different. In addition to that, also, well, write out, well, how are they different? Function, what is the difference function? On the left side, we have the cilia that you'd find in the respiratory epithelium. And because they're motile, they have the 9+2 configuration of their axonemes, and they have the dynein arms. Purpose is to sweep and propel stuff. Whereas the stereocilia and microvilli, they're non-motile. Their purpose is really for absorption. And both are made of actin filaments or microfilaments. If you don't know what you're looking at, also, because sometimes they may just give you um, a picture that has no goblet cells, no visible goblet cells, but you look for other clues like does this thing look like the nuclei are on top of each other, like there's many cells on top of each other? Are there goblet cells around? All right. Tricky, right? Because you can also see goblet cells in the intestinal epithelium, but note the differences in the lengths of these apical modifications. As we said, microvilli almost always looks like buzz cuts, and it's really hard to, on a regular H&E, to see the space in between the microvilli. Okay. I use this analogy today, like microvilli, because they're brushes. Think about the brush that you would use to clean the tubs versus cilia would be, like the strands of a mop. They're much longer. Okay. And then just some special considerations. For cilia, we have the non-motile um, variants with the 9 triplets. There's no central pair, no dyneins. Some of them actually are part of the sensory system. Stereocilia, think about pictures that have, oh, what is this? Well, what's in the middle? Oh, there's spermatozoa. I'm probably looking at epididymis here and not respiratory epithelium. It would be very unusual, okay, to see spermatozoa next to respiratory epithelium. And then we discussed earlier that microvilli, which you can find in the small intestinal epithelium, but also in the proximal convoluted tubule epithelium. Their purpose is they are for absorption, and microvilli have these glycocalyx or carbs covering the microvilli. And on a PAS stain, and believe it or not, these are both PAS stains. Again, it depends on the lab that does them. Look at the loop where the lumen is, that's where the apex or the apical portion of the cell is pointing towards. This brighter pink stuff here, that's the brush border of the proximal convoluted tubule. And on the right here is the tip of a small intestinal villus. And you can see that the periphery highlights this magenta line, and that's actually the brush border, the microvilli. And again, just to emphasize that on a PAS stain, it also highlights basement membrane. And we use a PAS stain, one of our favorite stains in renal pathology, to look at the basement membrane of tubules and to figure out, well, are there inflammatory cells that broke through and crossed the basement membrane and bothering these epithelial cells? Okay. So PAS stain is not just a special stain, but a very special stain in renal pathology. Okay, so just a study hint or study strategy. Organize your, your studying in tables. If you're confused about stuff, just or how things look, and you're confusing one from the other, do a side-by-side comparison. All right. Let's see. Where was I? I'm just all over the place. I'm not gonna. Any specific questions that I can tailor as far as my answers to. So we can, so we did a little bit of PAS stain. So how about, let's just look at H&Es, okay? I mean, some of these images are H&Es. Okay, let's just go back to this. I'm gonna go back to this slide. All right. So let me see if this actually will work. What cell is this? I mean, I'm covering it with my hand, but it's silly because I can't cover it for you. But this is a loose bronchial epithelial cell. Okay. And why might it be loose? Well, when somebody has issues with their respiratory tract, they can be subjected to a test, an imaging test called a bronchoscopy. And when we do bronchoscopies, one of the things that we do to also collect cells from the respiratory tract is we not only do we put in the scope, but we flush some saline into the respiratory tract. And then when that saline is sucked back up, it does drag some of these loose respiratory cells with the fluid. And that's what we can look at under the microscope to see if we're concerned about, say, a cancer, possible tumor in the respiratory tract. Maybe some of those cells can break off and get dragged into the saline, and that's what we will see under the microscope. This guy here is from the website I Love Histo. It's really cute, a cute website. It looks like a punk rock guy, right? With the what do you call it? A mohawk. But these, because it's a respiratory epithelium, bronchial epithelial cell, these are what is this then? These are cilia. And do you see this dark looking line here, or what looks like possibly just a dark line? How do I know? I'm just letting you know. That and plus, well, this image is of a cell that has little hairs on the top. Okay. But whether it's first off, it's really, really hard to sample cells of the epididymis. Really, really difficult. Difficult. Okay. But I'm also telling you that this is a bronchial epithelial cell. It's from the respiratory tract. Okay. Um, okay, you see this dark area here? That dark area is actually that's the basal bodies, like basal bodies side by side. That's where these guys are anchored on, okay, on this cell. And that's why they're up here because this part is facing the apical portion or the lumen of the respiratory tract, whereas the nucleus is sort of pushed down to the basal layer, okay? Why is the nucleus purple? Okay, purple or blue equals basophilic. So what does basophilic mean? Just break it down. The word means what? Basophilic. It loves bases. What loves base? Acids. So the nucleus have stuff in there that are acidic. Exactly. You guys are right on. And what's in the nucleus that's acidic? Deoxyribonucleic acids. Yes. Yes. The blueprint of life. Exactly. Um, then compare that to the cytoplasm or even these ciliated structures or these cilia. Why are these guys pink? So they're pink because they're taking up the pink dye. We also call it eosin. Eosin is the pink dye. That's why we call them eosinophilic. Okay. I appreciate the, breakdown of, well, it's basophilic, therefore it must be acid. It's not going to be true all the time. So that's why there was this one slide that we gave you to sort of a shortcut because there are things out there in our bodies that are not going to be existing on its own. Like for example, calcium ions are not going to be existing just in their own calcium molecule selves. They are always interacting with the environment. So there was a slide, and I forget which one it was where it describes if the structure you're looking at has a net something charge, then it would likely be basophilic or eosinophilic. Okay. So I'm trying to think where that is. Do you guys know which page? I think it was in the first lab slides. Look for it. Net. Maybe it was in the interact. Oh no, here it is.
What?
It is page 4, at least in the files um that we have. Let me get out of here. Page 4 of the first lab slides. Here you go. Components with a net negative charge, for example, nucleic acids, will stain with basic dyes, blue hematoxylin. Do folks have some questions? Okay. And then components with net positive charge, for example, proteins, will stain with eosin. Okay. And it's sort of hard to wrap around, you know, if you try to be, you know, so rigid with the rules when you know, don't think that, oh, just because it's basophilic, it must be an acid. So for example, what has a so calcium, what's the charge of calcium? Just the calcium ion. 2 plus. 2 plus, that's positive, right? Would you expect it to stain pink, at least in tissue? Let me tell you, when we see cells that sort of are dead or it's leaking calcium, what we actually see as far as, oh, there's possibly a leak of calcium there, is that we see a streak of blue. Why is that? Because calcium ions don't exist in isolation in tissue. It interacts with its environment. So in those areas where calcium ions that have leaked out of cells, they then encounter other molecules and structures around them that in that area, they may actually have a net negative charge and therefore may appear blue on an H&E stain. Okay. But in general, for your purposes right now, on an H&E stain, here again, what epithelium is this? Buzz cut. Small intestine. I got you, SI. You guys doing the abbreviations? At least I figured it out. Okay. So you see that the nuclei are blue because they have nucleic acids. Then you have sort of a mix, sort of like a lavender hue to the cytoplasm. Why might that be? Why is it not completely pink? I'll get back to you, Jay, but it is not because of lipids. I'm talking about just the cytoplasm, the area that's not occupied by the nuclei are down here near the basal edge of the cell. And this is what polarity is, right? Epithelial cells have polarity, meaning there is one side is different from the other side. The apical side already is different from the basal side. The apical side have these special modifications of their surfaces that is not present in the basal side where it attaches to the basement membrane. And why is this nucleus sort of pushed off to the basal side? Well, because there's stuff in the cytoplasm that's taken up this part of the cell. So why is that not completely pink? Honestly, so there's ribosomes too. What are ribosomes? That's a good. What ribosomes exist in the cytoplasm. Ribosomes are made of what? RNA, good. Could there be other things in the cytoplasm that say, could also be basophilic? Of course. There could be some stuff in secretory granules that can definitely pick up blue stain or the hematoxylin. I saw a question earlier. Do pathologists, well, H&E, hematoxylin and eosin is the gold standard of, like, it's our first first view of the tissue. That's this gold standard stain. We look at it first, and then if we're interested in exploring other proteins or other structures, then we do other special stains. All right. So Jay mentioned lipids. Well, in these images, we don't really see lipids or, isolated lipids, but how would lipids look like on an H&E stain? Why are lipid-filled structures, why do they look clear on an H&E? Fatima said they get washed out. Why? Yes, they absolutely get washed out. During the steps of preparing the tissue for preparing the tissue if you're going to embed it on a paraffin block and then cut it and put it on a slide. So just to also, I mentioned earlier, that depending on the lab, whether they can afford more hematoxylin, goblet cells are not going to look completely pale or white. Okay. This one is nice because it really highlights even some of the whatever granules, the mucin granules inside inside this goblet cell. Okay. So it varies it varies with the lab, but in general, cells that contain mucin are going to be paler, much, much more paler. Okay. Let's see if we can find some other stains here. So going back, why are lipids clear? Well, in the preparation of tissue for microscopy, at least light microscopy, which step here might possibly leach out the fats from fatty structures in the cell?
Grace, really quick, can you make me co-host so I can mute
Oh,
some?
yes. Sorry, yes.
That's okay.
Yes. Yeah. Well, what happens when you squeeze some lemon onto some fatty pork belly? I don't know. But I do know. But really the alcohols are gonna leach out some of the lipids, if not all of the lipids, during the preparation of tissue. Okay. And when the structure isn't there, it's not gonna pick up any stains. Okay. And in the quiz, and I hope you guys do the quizzes, the Canvas quizzes before class. That way you can check your grasp of the material during class, or even bring up questions to clarify if you miss stuff in the quiz. It's not graded, but you should do it because it's extra practice. And we encourage you to do it before class because guess what? I'm sure you guys as TAs, wherever you are, just generated a bunch of slides of extra questions that you can do. So really, and please just do those Canvas quizzes before class. Okay. Um, there's a one question. The first question in quiz 1. What might be a special stain that we can use in the lab to highlight intact fat in tissue? So fat that we haven't processed in this manner here for microscopic study. So just you collect something and it has fat in it. What might you use? What kind of stain might you use to highlight fat globules? Good. Sudan black. What else? Oil red O. We use oil red O for frozen tissue. I think other labs use Sudan black. So those are types of tissues that have not undergone the dehydration process. So it hasn't been immersed in alcohols. So you still have the fat in the specimen, and therefore you can assess the presence of fat by using special stains like oil red O or Sudan black. Okay. All right. Now we're talking about stains. And remember, there is a nice appendix. The last part of the Junqueira textbook is going to have a list of all the special stains, not all this, most of the special stains that we use in the lab. Are you guys able to access those or do you need me to pull it up? Do you see my screen of the textbook?
Yes.
Okay, so good. You see this as appendix, light microscopy stains.
Yeah.
Okay. Uh, one edit I would do is actually have example pictures. All right. So let's go through. We just talked about H&E. Toluene in blue we typically use for other types of microscopy. We use it for electron microscopy, at least. I'm not sure what para rosanilene toluene blue, but maybe toluene blue is just the shortcut for it. Mallory trichrome is important because it really brings out one of the most prominent colors on a Mallory trichrome is the blue color. And let me just see if I can find an example in our slide set. So on a Mallory trichrome, you read at least that, and there are different types of trichromes. There's a Masson trichrome, Mallory trichrome. Different people have made their different formulations of the trichrome. But the point of this really is this, to differentiate it from these guys and to differentiate it from these guys. So nuclei will stain purple or, some kind of dark gray at times, depending on the lab and the technique. Cytoplasm, keratin, and red cells are going to stain bright red or orange. And then collagen will stain bright blue or light blue. Okay. And you know about PAS stains. Purplish pink is another term for magenta. And sometimes they can just be, very, very like fuchsia color. All right. We have other stains. Oh, I have not completely logged in or it kicked me off. But in that first question, there are different stains. We also have stains for elastic fibers. Um, there are different types, but we'll pick up on those a little bit more for next week when we delve into connective tissue. Yes, Orcine and Resorcin. Perfect. Yes. Okay. Any questions while I look for a trichrome stain? Van Giesen is actually a stain for elastic fibers. And
That's usually used more for vasculars, for vascular tissue.
yeah, exactly. The elastic fibers in vascular tissue. And we use that to see whether, processes like atherosclerosis has completely chewed up or destroyed the structure of the elastic fibers within the walls of these vessels. Let me see if I can. Oh, all right. Anybody, someone had a question, show how to get to this page. Oh, Sterling, it's going to be, if you are on the online version, if you go to the online version of the textbook, it's going to be at the very bottom. There's going to be a list of all the chapters at the bottom. It says appendix, light microscopy stains. So this is a trichrome stain of a liver biopsy. Do you notice that there are cells that have red cytoplasm? The nucleus look, I guess, dark purple. And then these areas here look blue or shades of blue, dark blue, light blue. So the areas with blue or dark blue, those are areas with collagen. This is a trichrome stain. Okay. Blue highlights collagen because these are areas of this liver biopsy that has started to scar. Okay. And you can see that there are these blue lines sort of encircling each of these cells. And we use this patient, I'm going to tell you right now, has diabetes. Not quite cirrhosis. Cirrhosis will expect a different pattern of scarring, but scarring of the liver biopsy, like forming nodules, um, this is maybe like the early part of cirrhosis. But in this patient has diabetes. They are starting to form these scarring, really thin bands of scarring around each hepatocyte or each liver cell. And it's a particular look. Not every patient who has diabetes will have this change in their liver. But again, to highlight collagen that is in scar tissue, we use this stain called a trichrome stain. Because on H&E, this is just going to look very pale. And pale areas can either mean whatever's pale is either air or water, fluid or edema, or fat. Okay. It's important to, collagen is still important for structure, okay? But then again, there's some disease states where there's a lot of scarring, therefore a lot of collagen are going to be picked up by the stain, this special stain called trichrome. Trichrome is important because they really definitely make the blue pop or make the areas with collagen pop. Right. I am going to pause this and show you another. This is more of a lower magnification of this image. So same biopsy, but you can see the distribution of the blue now. Sort of it's almost coursing through, like infiltrating, quote, infiltrating around through each of these liver cells. But there's a lot of blue. Therefore, this liver is starting to feel really stiff, okay? In a normal liver, you will only expect it to be around the portal tract, so which houses the hepatic artery, the bile duct, and the portal vein, but not in this degree. This liver biopsy is not normal at all. Okay, because I gave some time to trichrome stain. I want you guys to see, where is that? As I'm looking, people can also ask questions. Oh yeah, this is a good one. One of my favorite pictures. This is, just blast it up here. What organ are we? Where are we right now? Kidney, good. Is this normal kidney? We'll get to it in the urinary system unit. So it's not normal kidney. Why? Because the glomerulus, they're the glomeruli, which are modified or very special capillaries, they sort of are scarred over. You see these large balls, yeah? These pink balls. Okay? And look, these are the tubules. And you can see that the periphery of the tubules are also looking very magenta. Oh, and look at the apical portion of these tubules are looking like a fuzzy magenta. Yes? So scar tissue will have a lot of collagen also, but also basement membrane have laminin. So this is, what stain is this, first off, since we have moved on from trichrome? This is a PAS stain, absolutely. PAS stain will highlight carbohydrates, as we mentioned. And in diabetes, this is a kidney biopsy of somebody with diabetes. This is called diabetic nephrosclerosis, meaning there is a hardening of the kidneys because of changes to the basement membrane because of uncontrolled blood sugars. All right. Not for the exam. I'm just giving you an example of why the stain is staining the things that they're staining. So in diabetes affected kidneys, the basement membrane is going to expand. And how we can visualize them is this is sort of an expansion of the basement membrane, and it looks like it's scarred over. And remember, basement membrane also has laminins, which have carbohydrates in them, and that's why they're picking up this magenta color on a PAS stain. That's all. It's just another example of a PAS stain.
I just wanted to make clarification because this question has come up a couple times in the chat. For
Yeah.
the organs, for the organ systems, folks, like for the only time that we really talked about organs were when I gave you some examples of organs that have a certain epithelia.
Yeah.
So there are some very particular examples. So for right now, just that. Once we get into organ systems, then that question, what organ is this, becomes fair game. Right now, only for the only based on the examples that I gave you according to the epithelial lining.
Yeah. We're just talking about epithelial lining. And I'm only showing this because I want you to see how PAS stains highlights the basement membrane, but also the brush border, but also areas that have expanded basement membrane material that is picking up the magenta color of a PAS. Maybe that was disconnect in my train of thought earlier, but let me see if I can actually, it's not so much annotate, but where is the pointer? I don't know where I find the pointer feature here. So I guess this is it. So this is a profile of a tubule. You see that I'm circling it with a pen, and you see that the edge of it is very brightly magenta. That's the basement membrane. The same here. It's outlining the outside of this tubule. That's the basement membrane. And it's being highlighted by the PAS stain. I know that this is a proximal convoluted tubule or part of a proximal convoluted tubule because the brush border is being highlighted here extra pink. That's all.
So in
That's
normal
just
H&E, you should not necessarily expect to see the basement membrane. Usually, for example, the most common place where you can kind of see a light eosinophilic region that is going to correspond to the basement membrane is usually on the trachea because for reasons we don't know, the tracheal basement membrane appears to be thicker than in other parts of the body. Otherwise, you really need PAS to visualize basement membrane.
Yeah. Let me see if I have any other stuff here. But okay, so I'm trying to think if there are any other stains that I need to talk about. So we did PAS, H&E. All right, don't forget the functions of the different organelles. This, yeah, so it's a great point, actually. Some labs can actually do two stains on one slide. And that is why it seems like I'm looking at an H&E, but the basement membranes are extra highlighted, right? Or these scar balls are extra highlighted. It's because it also has a PAS over it. Thank you. I'm sorry. I know it's not, I'm just giving an example. I shouldn't scare you guys, but let's see. I think I maybe was not highlighting or did not, I had the share paused. But yeah, this is the basement membrane highlighted by a PAS stain. So back to the H&Es. Let me just get out of this. Or back to the topics. Any questions with regard to the cytoplasm, nucleus, or the techniques? Oh yeah, techniques. We're back to the techniques. So, what other techniques did we talk about in class? And can you give an example of how might that be used? Like in So let's stick to maybe staining techniques. So why do we do stains again? Because first off, some of these stains, special stains like H&E, PAS stain, trichrome stain, Sudan black, Orsian, Resorcin, they interact with certain properties of the structure of interest, and that produces the particular color if they're present. So with Sudan black, that dye interacts with lipids. Therefore, what you see where there is lipid, it's going to have black blobs. Okay. For PAS stain, again, where there are carbohydrate-rich structures, you're going to see that bright magenta color. For trichrome, where there's very prominent collagen, you're going to see structures with a lot of collagen that's going to look blue on a trichrome. Okay. If we want to be specific, okay, let's say I am interested in looking at cytoskeletal components that are made with actin or, desmoglein, this protein that is found in desmosomes. What might I do to identify those? No, Sasha, no. We don't need to know about the particular plate. Anchoring support for stereocilia. You do not. Good. Immunohistochemistry. So immunohistochemistry really is, so if I'm interested in a particular protein in the cell, what I'm going to do is introduce that protein into a vessel, say it's actually an animal, that it's never been exposed to this protein, and therefore, once exposed, they're going to build antibodies against that protein. Okay. We will then collect this little animal's blood that contains all these antibodies that are specific to that protein that we're interested in. And we're going to use that as our detector antibodies. So we expose a piece of tissue when we're asking it, does this piece of tissue have this protein we're interested in? We will expose it to the antibodies that we grew in that animal, and that those antibodies will be, would attack or bind to those antigens if that protein of interest is present there. The second phase is actually, well, how do I know? How am I going to detect those antibodies that we grew in the animal that detected that protein? Then we have the second set of antibodies that are connected to a signal molecule, whether it's color, a chromogen that we would use actually on slides, or a fluorophore that would fluoresce if you're using fluorescent microscopy. If those antibodies can identify these antibodies that are tied up with the protein of interest, then the signal is going to go off. On a stain slide, they typically are, we use brown chromogens a lot. So if we're looking for things that contain desmin, we'll look for a desmin specific antibody. And on a slide, when we run that stain, it'll highlight structures that have desmin brown. Okay. Let's see. I know I'm a verbal person, but maybe I should show you guys associated slides with it. Okay. Yeah, you guys will be fine. The thing is, we can't put pictures on everything. So also understand how we describe stuff in text form. or talk it out with your internal voice as you read it. Well, where have I read this description before? Okay. Let's see. Important to know the types or the modes of cell transport. Transport. So you guys know what exocytosis and endocytosis? So know that.
So there's a question there. Diego asks, what is the difference between immuno and fluorescent microscopy?
Yes.
So
So
oh, go ahead.
go ahead. No, go ahead.
Sure. So immunocytochemistry is the technique used to label a protein. Fluorescent microscopy is the technique used to then detect what you've seen, what you've labeled on the slide. So one is to tag a protein, and the other is then to visualize if there's been that, if tagging has occurred, you can use fluorescent as a way to look at what you tagged.
Yeah.
Correct. Dye
Um,
visualization then. Correct.
exactly. So similar to using antibodies, but in when you're interested in looking at, say, DNA sequences or components of a chromosome, like looking at alterations in a gene, that's mainly how we use a lot of fluorescence microscopy. So instead of actually using antibodies, we use sort of these fluorescent tagged complement strands that would look for these particular sequences. And if it's found in the patient's chromatin, then the fluorescent signals would go off and we'd see those under a microscope. And oftentimes that's how we assess for, well, is there an amplification of this gene? Are there more copies of this gene in this person's cell? Because if it is, then this is the type of breast cancer that they have. This is how they might respond to this specific treatment that is developed for this type of change in the gene. Yes, so we just recently talked about the mechanisms of secretion. There were three main types that were highlighted, at least in the lab. One of them is holocrine secretions, or the holocrine mechanism, and how is that achieved? I'm literally running how I run class right now. I'm just gonna wait till you guys respond to me, while buying myself time to look for the slide. Yep, I think it's that. I think it's lab 2. Okay. Yes. So, and what's an example of glands that release their contents into the lumen via holocrine method or holocrine mechanism? Yes, sebaceous gland. So, there's this really good picture. Just why am I not seeing it today? Okay. Is it from this gland or this picture? Okay, maybe it's not that file. Okay. So sebaceous glands. First off, where are these cells replenished? Where are epithelial cells replenished? At the surface or at the bottom near the basement membrane? Yes, the basal layer. The basal layer is the location of stem cells, of our basal cells. Basal cells, sometimes we interchangeably call it, consider those as the stem cells. But in conversation, sometimes we interchange those. Okay. So the youngest cells are going to come from the bottom, and the older cells are going to be what you would see move up the layers. The cells at the very superficial layers are the oldest cells. In skin, those would be the top part, the top layers that would have already kicked out or have no more nuclei. Okay. In sebaceous glands, those would be the ones that would eventually disintegrate and release all the contents. So the waxy esters that sebaceous glands produce, that's going to be released. And that's what produces the sebum on our skin. Okay, that the sebum is what is released. The contents of the entire sebocyte or sebaceous cell, that's what they completely released. They completely disintegrated. Okay. What are the other two methods that we learned of or we discussed today? Good, apocrine. What's an example of apocrine secretions? So what epithelium performs apocrine secretions? Good, mammary glands. And how is that different from, say, a merocrine or merocrine secretion? I could have sworn it was on this slide set, but I cannot find it. Okay, so, oh, you know what? It was in the interactive session slides. That's why I couldn't find it. Okay. So, well, first, merocrine secretion, what is the mechanism of content release? Exocytosis. Good. How is exocytosis achieved? What happens to the vesicle that contains the stuff that you want to spill out into the ducts? You can see it here. So that particular vesicle, the membrane of that vesicle would fuse with the plasma membrane and then just release its contents into the lumen. There's no part of the membrane that is given up. It just releases the stuff that's in the vesicle that has fused with the membrane. Farah has a nice little mnemonic, if you will. Apocrine, a portion is gone. Think about the mammary glands. Apocrine, it is giving up a little bit of itself, like the mother is giving up a little bit of itself to the recipient of whatever it's giving up, right? So the apocrine secretion is, let's see, right here. You see the budding of a piece of the plasma membrane. So imagine this bud here has a little face, and then eventually it sees that it's going to end up like this, floating out into the lumen of the duct. All right. So this little vesicle has budded off, and that contains all the milk lipids, the colostrum that's going to be part of the milk. Okay. All right. What epithelium is this? It actually varies. You know, epithelium can vary in shape and size. So what may look flat one day, depending on whether it's actively proliferating or actively producing stuff, can appear cuboidal or columnar other days. All right. Classically, when we're talking about mesothelial cells, which are the cells, what kind of epithelium are mesothelial cells? What are mesothelial cells? I'm gonna look for that here. It's part of the one of the last, I believe. Nope, maybe not that. Yes, simple squamous. So mesothelial cells are what makes up the covering of our organs, as well as the simple lining that is covering the chest wall, the abdominal wall, the pelvis, the walls of the sac that contains our heart, okay, the covering outside the heart. So, and these cells produce this type of fluid that allows for the sliding of the two walls apart from each other in case the heart wants to beat and get bigger when it accommodates blood and then it pumps it out. And in case you want to breathe out and expand our lungs so that our lungs would slide past the inside of the chest wall. So it's slick, okay? That's what mesothelial cells do. Um, this particular image is not mesothelial cells. Let me just pause the share for that. I know people just got confused about it. Looking for the mesothelial cells in the slides. Here we go. This is not mesothelial cell. Oh no, this is a mesothelium here. Come on, share. Where are we? But these arrows are pointing to these cells here that look really squiggly and flat.
I think you're still on the, I still see your, there you go. Now it's showing. Now it's switched.
I'm looking at 3 screens here. Okay. Where are we? Yes. Yes. Now we are looking at simple squamous epithelium. And where are we? So when you're looking at images, always don't just look at where the arrow is, look at the periphery, okay? Look at the surroundings. So it looks like, all right, this is the mesothelium. I'm telling you this is mesothelium, but where are we? So there are these pink things here. First off, this is an H&E slide. So what are these eosinophilic or pink stuff here? Good, connective tissue. What's connective tissue made of? Yes, dense connective tissue. A lot of collagen, perfect. What is this here? I agree. It's a blood vessel. How do I know? There are red blood cells inside. And look at the lining of this blood vessel. What kind of lining does it have? Does it have lining cells that look short as they are wide, tall as they are wide, or just flat? Good. So endothelium are simple flat cells, simple squamous cells. Okay. What about this guy here on the left? This is actually another blood vessel. It's a capillary because it has this one red cell inside. So are these guys here. And they're also lined by a single layer of flat cells. It's really small, exactly. This on the right, though, is thick with 2 C's. Okay, and you know that you are in the lungs. Why? What is this business here? Exactly. This is the alveolar septa, or the wall. And that's a clue to tell you where am I? Um, I'm on the opposite side of this, and this just looks like nothing, but what these arrows are looking at looks like these really, really flattened cells with really flat nuclei. So this must be the simple squamous mesothelial cells. This is exactly visceral pleura. Good. It's the lining covering the actual lungs, the actual viscera or the organ. Okay. All right. Good job. Okay, let's test you guys. If I put a trichrome stain on this image, what will these guys look like? Who votes? Oh, I couldn't even do a poll. Be fancy here. No, I'm not doing that. It's going to take a lot of time. So what did you guys call these pink wavy things again? They're connective tissue. Good. And on a trichrome stain, you guys already said earlier, because these connective tissue bundles are made of collagen. A lot of it is made of collagen, or a lot is, collagen. On a trichrome stain, then what would these guys look like? They would look blue. Excellent. Perfect. Okay. Let me see if I can do another example. What is this? Again, another simple squamous lining. It is, I know people get confused. It's epithelium, it should be on the outside. So we're considering the lumen of the blood vessel. Also, consider it, well, this is endothelium, okay? And how do I know that? These are the smooth muscle that makes up the wall of this vessel, part of the vessel that you can't see, all right? These are going to be loose connective tissue out here. And so on a trichrome, these would look what? These would also look blue, but because it's sort of, they're a little bit looser, they may just look light blue, all right? So this was also, this was mentioned, at least when I first asked them, well, what is this? And someone said, well, this is non-keratinized stratified squamous epithelium. It's tough. And I will try to make sure we don't give you like super highly magnified stuff. And if it is highly magnified, it's because it's an electron microscopy image. And we'll definitely give you some landmarks to give you clues as to where you are, or at least the orientation of where you are in the cell. Okay. This is simple squamous epithelium or endothelium. This is the inner lining of this muscular vessel. This is either artery or a vein. Okay. All right. We looked at this slide here. What is this business? And where are we? Yes, good. Yes, Cade. Only stratified squamous epithelium right now. Only stratified squamous epithelium can either be keratinized or non-keratinized. Where do we find in normal tissues, where do we find keratinized stratified squamous epithelium? Good. The epidermis of skin, which is what we're looking at right now. All right. Why is keratinization of skin important? Yes. So you see how these guys at the very superficial layer, they don't look like they have any nuclei to them. I mean, these are what we consider, oh, these are your dead cells. Yes. But they still have a purpose, okay? Because they are filled with what? Yeah, filled with keratin. And keratin is some waxy stuff. And so they provide insulate, well, pretty much prevents us from losing a lot of, a lot of water to the air. So it helps protect us from dehydration. It's an oily, exactly, exactly. All right, there's some oily stuff here. Okay, and what's this here? Connective tissue of the dermis. All right. If I slap on a trichrome stain here, what might these guys look like? Blue, excellent. So this is the kind of quizzing that you guys need to do to each other as you are going through these images. All right. If I slap on a PAS stain here, what might this part of this epithelium look like? Yeah, it'll definitely highlight the basement membrane. There'll be more magenta appearing. Okay, good.
So I guess everybody's forced to shower though right before. Yeah.
Okay. All right, what about this guy here? First, and if you remember, Dr. Vidal said in the lecture, remember that you classify the epithelium based on what the most superficial cells look like. All right. These most superficial cells, what is their general shape? Flat, good. Therefore, it's squamous epithelium, and there's a bunch of layers, therefore it is stratified. Okay. But do you see that these top cells or top layers still have nuclei? Therefore, these top cells are not going to be dead. This is just an artifact. Sorry, when we say artifact, it just means this looks like it just separated from this part because of processing, and we weren't able to wash off this bit here. And remember, we're looking at these slides in 3D, so don't mistake this as not as a keratinization, because if you look at a cell, maybe the nucleus where the plane where they cut this particular cell, these cells at the top, the nucleus may not be in that plane of sectioning, of cutting. That's why it looks like they're just empty cells. Okay. Is Bella what the darker projections of cells you mean here?
Yes.
So this is going to be, oops, excuse me. So this epithelium is actually going in and out. It's sort of the connection, how it connects to the connective tissue underneath is sort of like this, just as skin is also connected to, or the epidermis is connected to the superficial parts of the dermis like this. So there's going to be a waviness to the bottom part of these epithelial layers. So it actually adheres to the stuff below. Okay.
So in between
Thank you.
them, it would be
Epithelial
connective.
ridges.
So in between would be like the connective tissue, and we just can't see it because of the cut.
Yes. So in this part, there's going to be this stuff here. It just goes in and out. Right.
Okay,
There's also
thank
some
you.
capillaries in here too. So I don't know if that skin image will have it. No, we are too high on that. But where might you find this type of epithelium? Where in the mouth? So your buccal mucosa, yes, esophagus, the vagina. Which part of the cervix? The exocervix or the ectocervix that is in continuity with the vaginal canal. Good. Because once the inside of the cervix or endocervix, that's a completely different epithelium, right? So the outer part of the cervix that is readily available through a colposcopy or a pelvic exam, okay, that is what we see. That's going to be covered with stratified squamous epithelium, non-keratinizing. Okay, at this point, we're talking only about normal histology, okay? That's right. Okay. Because clearly there are going to be some changes to the epithelium if it's subjected to irritation, whether that's mechanical or chemical irritation, okay, or irritation created by actual infection. All right, so there are going to be changes to the look of the epithelium. So epithelium, I think we briefly touched up on that in this, I think this week. Epithelium will respond by when it replenishes itself into a different type of cell or epithelium that will protect it from whatever is irritating it. Okay. So in the respiratory tract, say somebody is a chronic smoker, it's exposing all those nice pseudostratified columnar epithelium with goblet cells and cilia to a lot of caustic chemicals that are coming in from the smoke or inhaling soot. We do live in Great Falls here. We definitely have like a smelter. There's it does generate smoke. There's a smokestack. Or if you live in, we're going to talk about it in the spring, houses that have a lot of chimneys and you're inhaling all the soot from, having a fire, turning on a fireplace. So all this irritants that we are inhaling that's going to irritate the epithelium too. And so what might be a protective epithelium to smoke? Yeah, so so you'll actually see this process called metaplasia. Metaplasia is just a way for one cell to replace an irritated cell with a different cell that is protective. So cilia tends to stop functioning or becomes dysfunctional when it's exposed to smoke. So in the pathology lab, we sometimes get biopsies from the trachea or the nose of babies, from babies, because the parents bring these babies in and say, my kid is not, has always had a cold. It's not recovering. We don't know what's going on. We look at these biopsies under electron microscopy and we look at the cilia and we see whether they still have their 9+2 configuration of microtubules. And so it's very rare, but I'm lucky enough to actually see two cases in my short 1-year fellowship, two cases where the cilia from the biopsy looked really wonky. It did not have that 9+2 configuration. The one case did not have the central pair. Some cases did not have all 9, only had 7. A lot of the cilia did not have the 9+2. And so in those cases, it was supportive or suspicious for a condition called primary ciliary dyskinesia. So something happened that um, led to the not or the undevelopment or underdevelopment or wrong development of the cilia, even, before the child was born. But most often, like 99% of the time, nothing was wrong with the cilia. And so what we can only say in our report is the cilia that was biopsied and that we saw under the microscope, they all looked fine. This kid probably was exposed to other environmental exposures that caused them to not recover. And then we find out that the parents are chronic smokers and they smoke in the house. And so, um, at least in the cases that where we signed out as, oh, this is secondary ciliary, meaning it was caused by something else. It's not caused by the actual embryologic from embryologic development of the cilia. So we get the samples from the pediatrician. They send it to the lab, and then we process it so we can look at it under the microscope. We personally do not pick it out of the babies, so it's the actual pediatricians. Okay, so a protective type of epithelium is stratified squamous. Okay, and so you will see some stratified squamous epithelium in areas of respiratory epithelium that has been subjected to a lot of caustic chemicals, like such as in cigarette smoking. Um, and I saw earlier, why do we tell you guys these? Because over time, if you don't remove that exposure, that chronic irritant, cells that have to keep dividing to replenish itself, eventually will also be exposed to damaging um, substances that will affect its DNA. And as these cells keep proliferating, and if these changes in their DNA also include, well, I'm going to ignore the cell cycle checkpoints. I'm just going to keep dividing and proliferating despite these checkpoint proteins telling me, no, I'm not supposed to. I'm going to ignore that because I'm a mutated cell. That's when we get cancer. Okay. Um, and we're teaching you histology up front because it's actually very hard. It's harder to see and recognize normal tissues versus diseased tissues. But also, you're not going to know if a tissue is diseased if you don't know what normal looks like. Okay. We went through stains, secretion mechanisms of secretions. Also remember to take to remember all the clinical correlates that we discussed in class. Last week, was it last week or this week? So last week we talked about the cell cycle, but also, we talked about mitosis briefly and meiosis to know the difference, the events that happen during each phase. What happens when you get conditions like, I believe one of the clinical cases is, oh, let me pull that up. That was lab, I think it was lab 1. I didn't know if you guys were able to go through it in class. Sometimes I'd run out of time. Okay. Remember this case? Although this is sort of a bridge between topic 1 and topic 2. So a 28-year-old man, they've been trying to conceive a child, no success. Went to fertility specialist, found that the low sperm count, in addition to slow, motility is not great. Okay. Before performing the sperm analysis, good doctor, got really good history. Found out that there's chronic mucoid rhinorrhea. Rhinorrhea just means very like runny nose or diarrhea of the nose. Sorry, rhinorrhea. Rhea just means a lot of secretions, I guess. Repeated sinus infections, sinus headaches, ear infections. Okay. Negative for cystic fibrosis, because that's also a condition that, where there's a lot of buildup of thick mucus in the respiratory tract. And then we entertain these different types of conditions that could explain what's going on with this patient. All right. And then, yes, okay. Motile cilia are found in different places. Oops. In the fallopian tube, aka the oviduct, that is responsible for moving along the egg that came out during ovulation down towards the uterus. And then remember, the sperm have the flagella that have the microtubules, the 9+2 microtubule axoneme structure. Also in the epididymis, the efferent duct of the vas deferens, and then nasal cavity as well. So we get the biopsies from the babies like around here or maybe up down here, as well as the trachea. So this is just a review of the normal motile cilia microtubule 9+2 axoneme pattern. And that's what I just described when we looked at these biopsies. If we suspect primary ciliary dyskinesia, they're not going to follow these microtubules, are not going to follow this arrangement. We'll either not see these guys in the middle, or there's only 5 of them, or this is not here and there's 6 of the doublets from the outside, or there's actually absent, the dining arms are not there. All right. And in this case, what's wrong, or what do you notice about these CT scans? So first off, how do you read a CT scan? So the patient is actually, when you're looking at it, imagine that the patient's feet is actually on your face, okay? Or it's pointing towards your face. And when you're looking at these images, the patient is lying on their back. Therefore, their right side is where the R is on the picture. All right, so If you're pointing at it with your hand, you're pointing at it with your left hand, but it's the patient's right side. Okay. The right image is interesting because if you look at it, what are the placement of the organs? Where do you typically see the liver? The liver, the giver, is typically on your right side, right? Correct. And what's this organ over here? That is a spleen, and that usually is on the left side. What about this guy here with the black bubble? This is the stomach. Esophagus is more midline. Okay. So there is a flip of the location of these organs. Okay. Cilia are there too in the, I think the nodal cilia during embryogenesis to make sure that these organs get placed where they ought to be placed. All right. And if there's something wrong with those cilia, then you get a condition like situs inversus, inverted sites. Okay, the place where you expect things is going to be inverted, it's on the other side. And if you look at this, so the cardiac apex is actually pointing to the left side, it looks like, but in anyone else who does not have situs inversus, the apex of your heart should be pointing towards the right. Okay? Or did I get that mixed up? I think it's... So just some findings. All right, there you go. Situs inversus chronic sinusitis, bronchiectasis. Bronchiectasis just means widening of the bronchial passages. That's what ectatic is, or ectasis. Well, we talked about microtubules, and we definitely took time to talk about it in class. What do you think? Is it going to be on the test? I don't know, but sometimes we take time to talk about it in class because we think it's important. All right. What else? Oh yeah, here we go. Here are some slides or other slides. Nice side by side. These are sections of pancreas. If you just stain it with hematoxylin, it just looks all blue. With just eosin, very pink, but combine it, you can differentiate where, these structures are together. So the nuclei pops up and they look more blue, whereas the cytoplasm filled with zymogen granules are going to look more pink. Okay. Oh, this is beautiful. I thought I was going to have to do this, but it's already here, courtesy of our faculty, Dr. Vidal. I'm a fresh faculty, so
Oh, these are actually Dr. Bruno.
okay, Dr. Bruno. There you go. Thank you. I will, if he's not here, I'll personally thank him. So this is great, side by side, what the stains look like. So connective tissue stains, the stuff that is really being highlighted by like blue, the connective tissue that is very blue are the ones that have a lot of collagen. Okay. Elastic fibers are a type of connective tissue. So in this one, this is an elastic von Giesen or EVG. These fibers, these elastic fibers are appearing black in this particular stain. Silver stain also looks black, but they're specific to reticulin fibers or type 3 collagen in this particular image. All right. So there you go. This is the visual representation of the appendix of the textbook. PAS stain, mucins, as well as the glycocalyx covering the microvilli. We have another type of mucin stain, the Alcian blue. Musicarmin would actually produce a reddish orange color if we see cells that contain mucin. So there are different types of mucin, as we mentioned before, and some may not necessarily pick up some types of stains, and that's why we use different types sometimes. Yes, thank you. Thanks, Dr. Vidal. Yes. So what was that one? Cartaginer syndrome? Yes. What was another syndrome we discussed earlier in this lab? All right, pemphigus was this week's. Perfect. And what was the pathophase for pemphigus vulgaris? Good. Antibodies attacking the proteins that comprise the desmosome. Perfect. The mucicarmin, it's mucin. So these are mucins, mucin stains. Therefore, it's mucus producing cells in a mucicarmin. It's in the name. And it's carmine because the carmine stain is, I think, the orange-red pigment. Okay. Sudan red, it's in the name red. It would stain tissues that have intact lipids. So these are typically used in frozen tissues or fresh tissue. We don't use this on tissue that we've already processed, dehydrated, embedded. It's good, the lipids have already washed out typically. Okay. Prussian blue is another stain that highlights iron particles. Okay. And this is a liver that is full of iron. So this is actually a patient with hemosiderosis or somebody who's just had a lot of transfusions in their life. So they tend to build up a lot of iron stores in their liver cells. So this stain is bright blue because of the amount of iron they have. And von Cossa stain is to highlight stuff with calcium. So calcifications. It's a nice stain to use, okay? Okay, I love these slides. I have to give them more props. Oh yeah, going back because I got distracted by the pretty slides. It was around the cell cycle, just to, I guess, review that part. This one? Nope. Okay. It might have been in the interactive session. Okay, we did PV. What else did we discuss after pemphigus vulgaris? There was another blistering condition, and that one was bullous pemphigoid. Okay. And what was the target protein or target structure in bullous pemphigoid? It would be the hemidesmosomes. Good. Okay, so know the difference between those. What other conditions? Yes. The drumstick feature. Let's see. Am I giving away too much? I mean, I'm just directing them that this is giving them away too much. Yeah, I'll give you guys, yeah, no. That's why yeah, whatever. Okay. So in this particular individual, there is this little nub of chromatin that's jutting out of the main lobes of this nucleus. So this is a neutrophil. Yes, this is a bar body. And what is the bar body? It's the inactivated X chromosome. Good. And in this particular nucleus, okay, they are sometimes seen as these little areas of heterochromatin in the periphery. Okay. Here it is again. There you go. I even highlighted it for you. And when might you find bar bodies? Which conditions? So I would have to correct Joey, it's not necessarily intersex, because intersex individuals may actually, I mean, they have the chromosomes, but there are some like hormonal signaling molecules or structures that may or may not be there. It has nothing to do with their chromosomal makeup. So bar bodies can be seen in individuals that have more than one X chromosome. Okay, more than one X chromosome. And we talked about this particular condition, the Klinefelter syndrome, which this is a karyotype. It's kind of cool if you ever get an opportunity to get your karyotype done, do it, frame it. I framed mine, though I can't find it anymore. But we do this in constitutive genetics. If we are concerned about perhaps the fetus will have, based on findings in an ultrasound, if we're concerned, we'll do constitutive genetics, run a karyotype of the amniotic cells and produce this picture of all the chromosomes of the fetus. And really, for the most part, we're interested in these chromosomes here. If there's a third copy of chromosome 21, then that's trisomy 21. There are other conditions that may not be compatible with life. And that would just be extra copies of other chromosomes. In this here, we should only have one copy of the X chromosome and then one copy of the Y chromosome, but this individual has two copies of the X chromosome. Therefore, based on the Lion hypothesis, one of these chromosomes have to be silenced. X chromosomes have to be silenced. And that silenced X chromosome is what manifests as bar bodies in these cells. Okay, that's a neutrophil. And in this one, this is I think a buccal mucosal cell. And they saw these little blip of heterochromatin in the periphery of this nucleus, and that's the bar body. Okay. People can be mosaic. An individual can have actually XX and XY cells. And therefore, how many bar bodies might that person have? I guess that's a trick question. Okay. But yes, so typically it's the number of X chromosomes minus one. That's the number of bar bodies you might have. Due to non-disjunction events. So somebody can actually have three X chromosomes and one Y chromosome, or three X chromosomes only. And so you just count the number of bar bodies would just be, again, number of X chromosomes minus one. All right, cool. Heather picked it up. Yes, and folks who are chimeras or mixture, some cells have just XX and some cells actually have XY. Therefore, the cells that only have an XX will have one bar body. Okay, that's a little complicated, but okay. For mosaics, yeah, I guess it would depend. It would depend if it's two different versions of an XX cell. Yeah. But if it's if some cells are XX and some cells are XY, then the bar bodies would be only applied to the cells that are XX. This is a more complicated, advanced conversation, but okay. Areas where non-disjunction might happen, it can happen as early as up here in meiosis 1, anaphase part of meiosis 1, or it can even happen like later on here. Okay. And you know the answer to this, should know the answer to this already. Good. Okay. Let's see. What other conditions do we talk about? Oh yeah, epithelium. We mentioned the two bullous diseases, pemphigus vulgaris and bullous pemphigoid. I think we briefly discussed celiac disease also. I don't know. Are there anything else? So other stuff, know the different functions of the cell organelles. They know the organelles and their function. If it was mentioned in the lecture, if you read it in the required reading, if it was in the slides for the interactive session or the labs, it is all fair game. All right. Okay. Any specific questions? I noticed the time is already 7:11. And again, I'm gonna stay until everyone's questions are satisfied, but I definitely would want to have a hard cutoff at 7:30. Quiz question that had the options of anaphase 1 and anaphase 2. Well, I think you can send me a screenshot of that question, Diego. So I'd have to see it, but there are definitely different phases during meiosis where non-disjunction can occur. If you had to ask Amy, the answer is what? So what's the answer to this question? Yes, it's B. Good. This is where it happens, where there's an imbalance or imbalance separation of the chromatids during meiosis 1, anaphase of meiosis 1. And so what ends up in the gametes, the haploid cells would be, three copies here. All right, and this one did not get these copies, an even copy. So it's always good to review normal versus, the altered event. Okay, so this is the expected on the left side, how the chromatids should be separated in the formation of these gametes or these haploid cells. But in non-disjunction during meiosis 1, this is the distribution. Okay. Let's see if I can answer that question. Is there anything else that I should cover? I should know because I did not get the most final version of the test yet to test, but
And I think for as far as we're concerned, I guess just so that we can set expectations, so the exam questions compare. Yeah, so I had this discussion with my students, but I want to make sure that I know some people have already left, but I want to make sure that everybody from all three schools are aware of this, right? So this always happens every year where students come up to us and go, wow, that test was so much harder than the clicker questions or the discussion session questions that we get, right? And the thing you guys have to realize is that when you come to our sessions, your focus is very narrow, right? You're just focusing on histo. You know that you're going to expect histo questions. And again, more specifically, you know what the topic of discussion for the day is going to be. So there's no real surprises there. When you get to the exam, your focus is wider, and even more so now that we're doing module exams across different disciplines, right? And so that changes your perspective that's going to change your perception of the difficulty of the questions. But I guarantee you that all the clinical vignettes, all the typical questions that you saw in our clicker sessions and our interactives are going to be comparable to what you can expect on the exam.
yeah. And just a heads up, sometimes the exam software may not make things look uniform. So if you ever run into answer choices that are of a different font or a different shading than the rest, that doesn't mean we're trying to trick you. Choose the correct one. Don't get distracted by font changes or color changes, okay? Sometimes just the software doesn't succeed in unifying the look of the entire exam.
Yep.
We're not trying to trick you. It's going to be effective at distracting you, but we're neither giving you the answer nor giving you distractors. It's just a glitch in the exam software sometimes that you may see answer choices or certain text in a different font or different shade or different size. So, yes.
And I just want to add one other thing, right? A lot of students, when they come to talk about the exam, sometimes they'll say, "Oh man, I chose that one, but then I chose for this question, I chose this answer, but then I don't know why I changed it," right? So here's what I have to say to that. If you are answering the exam and you picked an answer choice, stick with that answer choice, right? And you are not allowed to change your answer unless you can answer the following question, unless you can fill in complete this following sentence. I am changing my answer from A, for example, to D because reason. Right? There has to be, you have to have a valid reason for doing that. If you cannot finish that sentence, right? Oh, I changed it because I didn't like the look. No, that's not. Or oh, I didn't trust. No, you have to have a proper justification for changing that. Majority of the times, what you went with will be the correct choice. Not
Are you?
always,
Yeah.
but if you start changing willy-nilly, that's when you start introducing errors. And by the time that you change it and you submit that exam, that becomes fine.
Yeah. And I was practicing on one of the versions of the exam today. I noticed also when I clicked on the answer choice, either it's gonna, I don't know what software you guys are using, but when I clicked on what I wanted to choose, um, sometimes it just clicked on the I and it struck out that answer, which meant the test did not officially registered my answer as that answer. So ensure that when you choose it, it actually will have a check mark when you choose the answer. Okay. Because if you're doing it really fast, and I don't recommend it trying to rush through the exam, ensure that you read the question carefully, ensure that you pick the correct answer because some people said, "Oh, I saw that answer. I swear I chose that. I don't know why I ended up choosing something else." That's an instance of people just, not making sure that they chose the right answer or did not click it and ensure that they clicked on the right answer. Another is that I'm pretty sure you guys are not going to be sleeping, or, I don't recommend not sleeping before the exam. But if people are tired because they're up late, that's also going to increase the chances of people doubting themselves and not trusting your preparation. Okay. Trust your preparation. Resist the urge to change answers. And if you have to come up or spend as much time, if not more time, trying to justify your alternative answer that you're trying to entertain but cannot 100% be sure about, maybe just stick to your original answer. Okay. That was the thing that I remembered, the main advice that I had to follow when I took my MCAT for the first and only time, um, to resist changing my answer. And I'm a perpetual answer changer, okay? But during that time, I did not do it. So, and then I ended up doing pretty well. So trust your preparation. Pinch yourself if you are getting the urge of changing the answer, but only until you do what Dr. Vidal said. Have a good reason for changing your answer. Okay. Any other question? It's 7:20. I don't know what else I can talk about. I think we prepared you well enough. Um, again, I remember the first exam last semester, it was what, like 6 weeks worth of stuff?
Yep.
Yeah. So
6 weeks worth
this
of
might be
stuff,
helpful.
and we didn't test until the 9th week.
Yeah. It's a little intense to get a test already, like just barely 3 weeks in, but this might be helpful. But again, good luck
Yep.
with your preparation.
You'll do fine. You will do fine.
Yeah. Again, email me questions if you forgot to ask questions or you come up with questions later. Yeah.
All right.
Thanks for